Good afternoon, everybody. Today we are going to start the new topic, biotechnology. This 12th topic, biotechnology. In this topic, we shall study about the two terms or two concepts are categorized into this biotechnology. The first term, it is some biotechnological processes and its application. And there are several applications of biotechnology. It is one of the applied branch of biology, and it is a various uses of this biotechnology. This biotechnology, it is a processes which takes place by using the microorganisms, and these microorganisms are some enzymes or some molecules. They are obtaining the different products, and these products are useful to human welfare. This one term, the first term, what is a biotechnology? This biotechnology, it is a scientific definition of this biotechnology. It is an application of scientific and engineering principle to the processing of material by biological agents. This one of the branch of biology, and it is a applied biotechnology. It is a applied branch of scientific and engineering principle to processing of a material by using a biological agents to provide good and service of the human welfare. This is a definition of biotechnology. It is a particular definition of biotechnology. This biotechnology, it has been broadly classified or broadly defined. The biotechnology is a development and utilization of biological forms, and they can produce the products, and these products is useful to human welfare. And this biotechnology, it is some application or some principle or genetic engineering is involved. It is the most important branch are involved in this biotechnology, and also some microorganism, means like a biological agents is involved for this process. Means microorganisms like bacteria, they can using fungus. This type of microorganism are involved for this biotechnology, and there are various uses of these microorganisms. And some uses of this biotechnology, some microbiology, it is one of the technology or one of the branch is also important for this biotechnology. The another genetic biochemistry as well as chemical engineering and mathematics, statistics, computers and industrial processes or industrial biotechnology is very important. Industrial purpose, this biotechnology is very important. And also some biological agents. Biological agents means that as the plants and animals, they are involved for this process, means plant cell or animal cells are using for this biotechnology. And some microorganisms or some enzymes, they are also useful for this technology. And this biotechnology, it is one of the important branch, and some terms or the development of this biotechnology into the some growth. They are occurring the two bases or two phases of biotechnology. One is a traditional biotechnology and another is a modern biotechnology. The traditionally, this biotechnology is very important. Traditional uses of biotechnology, for example, it is based on the fermentation process. This fermentation process is involved for traditional uses of biotechnology. Fermentation process means this fermentation process involves the various products like there are some example for fungus like yeast is mostly important for the fermentation process. This Saccharomyces cerevisiae, this one of the fungus are Saccharomyces cerevisiae. This one of the fungus like yeast. This yeast is using for the fermentation process. This microorganisms is also involved for the production of the various products like some preparation of some curd, ghee or vinegar, cheese, wine production, and these are different products are also produced with the help of these microorganisms. Means some bacteria is also important, or these microbes also used for the fermentation process. And traditionally these bacteria or fungus, they are using purpose for, they are useful for the various means in became an art of the kitchen of the Indian houses. They are delicious food material are also produced with the help of this bacterium or some fungus. Fermentation process is also including for this traditional biotechnology. Traditionally these are the some importance of biotechnology. And some new modern biotechnology, another one branch or another one terms is a modern biotechnology. And this modern biotechnology means there are also some manipulation of the gene, means RDNA technology is also important for, or engineering techniques are using for the some microorganisms are using, or these RDNA technology are also important. Recombination DNA technology are using this modern biotechnology. And this RDNA technology is studied by 1970, and later the 1973 Stanley Cohen and Herbert Boyer, these two scientists also studied or established the development of rDNA technology. This recombination DNA technology, rDNA technology, means manipulation of the gene for another cell. And this Stanley Cohen and Herbert Boyer, these two scientists also combination of biology and production technology, and they are based on the genetic engineering, and they can evolve the new terms or new biotechnology is a modern biotechnology. Means these are the one important concept or one important term. rDNA technology is using the modern biotechnology. There are some features of biotechnology. The first one, these techniques are also capable for the scientific change of the products like the rDNA, means recombination DNA technology, as well as some PCR, polymerase chain reaction, PCR techniques. Using these PCR techniques of this biotechnology, there are some important major feature of the biotechnology. Using rDNA technology means specific gene are insert from the cell. These are recombinant DNA technology, or some uses PCR technique, polymerase chain reaction. This technology means polymerase chain reaction. This technique are also using for biotechnology, means multiplication of the gene, means multiplication of gene in vitro condition. This PCR means multiplication of a gene in vitro condition. These in vitro conditions, these PCR techniques are using, means multiplication of the gene, particular gene, they are multiply within a short period, there are many copies are produced, and these techniques are takes place in vitro. And later the microarray as well as cell culturing process, cell culture. This another use of this biotechnology, cell culturing process, and also fusion of the protoplasm fusion. For example, fusion, culturing and fusion. With the help of this biotechnology, fusion or culturing means, for example, one of the plant like tomato. This one plant, for example, this tomato, variety is also developed with the help of this biotechnology. Means this culture or fusion, this fusion means the protoplasm fusion takes place, fusion of two cells. This one cell, the protoplasm is fuses with another cell, and they can forming the one of the modified plants, tomato or the plus tomato plus potato. These two plants, they are combined, means fusion of these two plants, protoplasm fusion of these two plants, and forming the new variety like a tomato. Means one plant is produces the two varieties or two types of the fruits are produces, means some half part tomato and half part the potato. This one variety is also developed with the help of this biotechnology. Means fusion or culturing of the cell is most important for this biotechnology. The another useful feature like the socio-political impact. In this biotechnology, these conventional industries like the pharmaceutical industries and also agro industries or some food industries, they are also benefit to human beings. With different these food productions as well as some other pharmaceutical industries, means this industrial purpose, this biotechnology is very important or useful technology. Later, next one concept, principle and processes of biotechnology. Principle and processes of biotechnology. Principle and processes of biotechnology. This biotechnology including the two phases, one is the traditional biotechnology and another is a modern biotechnology. And this modern biotechnology, it is based on two core techniques, and these two core techniques like the genetic engineering and chemical engineering. Two cores like genetic engineering and chemical engineering. This biotechnology or this modern biotechnology, there are some two principle or processes of biotechnology or core of biotechnology. They are including the genetic engineering and chemical engineering. These two terms is including the modern biotechnology and the genetic engineering. The first term, genetic engineering, means it deals with the alteration of genetic material. This term is used as alteration of genetic material. Like DNA and RNA. These two are the genetic materials, means the genetic engineering with the help of this biotechnology or one of the techniques, new techniques or modern techniques, they are involving the genetic engineering, this one of the branch. And this genetic engineering means the alteration of genetic material. The genetic material like RNA and DNA means manipulation of the genetic material from other cell. Means this genetic engineering, it is the manipulation of a genetic material toward the desired gene, and in a directed, they are predetermined way by using in vitro process, and this process takes place in vitro. This manipulation or altering the genetic material from other cell, it is important for the genetic material. And another branch, chemical engineering. This chemical engineering, it deals with maintaining the sterile environment. This chemical engineering maintaining the sterile environment for manipulation of variety of useful products, including this chemical engineering, they are including the some specific varieties like including the vaccine, antibiotic production, vaccines, antibiotics. This chemical engineering means some chemical concentration, they are sterile environmental condition. This processes takes place, the production of the vaccine, antibiotics, enzymes, organic acids, vitamins production, and some therapeutic uses of this chemical engineering. Means this chemical engineering technology is very important, very beneficial, and several production takes place under this chemical engineering, like the vaccine, antibiotic productions, or some organic acid, vitamins, enzyme production is also occurring in the chemical engineering. Now the genetic engineering means the manipulation of the gene, and this gene involves the repairing of a gene, of a defective gene, or replacing a defective gene for healthy genes or normal genes. This biotechnology is also including these two concepts, or they are principle of processing of biotechnology include the genetic engineering and chemical engineering. The genetic engineering involves the manipulation or alternation of the genetic material like the DNA and RNA. These are the altering the genetic material, and other is a chemical engineering, and several this production of the vaccine, antibiotics, these are several production is included in the